Most people who die of cancer do not die because their cancer was untreatable. They die because it was found too late. Let that sit for a second. The cancer was treatable. The biology was beatable. But by the time anyone knew it was there, it had already won. Here's a number that should change how you think about this disease forever. Pancreatic cancer. Caught at stage 1, survival rate 44%. Caught at stage 4, survival rate 3%. Same cancer, same biology, same molecular machinery destroying the same organ. The only difference? When it was found. And here's the brutal part. Most pancreatic cancers are caught at stage 4. Not because doctors aren't good enough, not because medicine isn't advanced enough, but because there is no routine screening, no test, no early warning system. You wait until you feel something. And by the time you feel something, the cancer has already had years to spread. This isn't just pancreatic cancer. Ovarian cancer, stage 1 survival, 92%. Stage 4, 20%. Liver cancer, stage 1, 31%. Stage 4, 3%. Colorectal, stage 1, 90%. Stage 4, 14%. The pattern is ruthless, and it repeats every time across dozens of cancer types. So why don't we just screen for all of them? The screening gap. Here's something medicine figured out a long time ago. Find cancer early, you save lives. Not through better drugs, not through more aggressive chemo, just by finding it earlier. The Pap smear, introduced in the 1940s, cut cervical cancer deaths by 70% over the following decades. 70%! Not because treatment got better, because doctors started catching it before it became dangerous. Mammography, colonoscopy, low-dose CT for lung cancer. These tests have limitations, false positives, access issues, overdiagnosis, but the core idea is unbreakable. Earlier detection saves lives. So why don't we do it for every cancer? Because building a reliable screening test is hard. A good screening test needs to be sensitive enough to actually catch early cancers, specific enough that it doesn't send thousands of healthy people into a spiral of unnecessary procedures, and practical enough to give to millions of people every year. That's already a brutal set of requirements for one cancer type. Now imagine meeting them for 50 simultaneously. Different cancers affect different organs. They express different proteins. They show up differently on imaging. A CT scan that finds something in your lung tells you nothing about your pancreas. A prostate blood test tells you nothing about ovarian cancer. Each cancer type has always needed its own dedicated detection approach, its own separate science, its own separate clinical trials, its own separate screening program. Developing and deploying 50 separate programs? Practically impossible. Which is why, right now, 47 of the 50-plus detectable cancer types have no recommended screening program in the UK. 47 cancers where the official medical strategy is wait for symptoms, then investigate. Waiting for symptoms is, for many of these cancers, waiting too long. But what if you didn't have to wait for symptoms? What if the cancer was already leaving evidence, quietly, continuously, long before it made you feel anything? What cancer leaves behind? Every cell in your body is constantly dying and being replaced. As cells age and break apart, they release fragments of their DNA into your bloodstream. This is completely normal. It happens all the time. It's called cell-free DNA, cfDNA, and it's just there, circulating in your blood, getting cleared out, constantly refreshed. Cancer cells do the same thing. They shed DNA into your bloodstream too, called circulating tumor DNA or ctDNA. And because cancer cells divide faster and die faster, they tend to shed more of it. This means if you have cancer growing somewhere in your body right now, traces of its DNA are floating in your blood. The cancer is invisible on any scan. You feel completely fine. No symptoms, no lump, nothing. But the molecular evidence, it's already there, in your blood right now. In your blood. The question is, how do you find it? Because here's the problem. That tumor DNA is a tiny fraction of the total DNA in your blood. In early stage cancer, we're talking less than 1%, a whisper inside a hurricane of noise. And even if you find a cancer signal, how do you know which cancer, which organ? Where do you even start looking? This is where something called methylation changes everything. The methylation fingerprint. Every cell in your body contains the same DNA, the same genome, but a liver cell behaves nothing like a brain cell. A skin cell behaves nothing like a blood cell. How? Methylation. DNA methylation is a chemical process. Specific molecules attach to specific points on your DNA and either activate or silence genes. Your liver cells have a precise methylation pattern that says these genes on, these genes off. You are a liver cell. Behave like one. Your neurons have a different pattern. Your immune cells have their own pattern. It's like a master settings file written directly onto the DNA. Cancer completely destroys this. When a cell becomes cancerous, it rewrites its methylation landscape. The genes that should be silenced, the ones that drive uncontrolled growth, get switched on. The genes that should be active, the tumor suppressors, the cellular brakes, get switched off. The cancer cell has essentially hacked its own control system. And here's the critical part. This rewriting is detectable. The DNA that cancer cells shed into your bloodstream carries the abnormal methylation pattern of the tumor, and different cancers rewrite the pattern differently. Pancreatic cancer has a different methylation fingerprint from ovarian cancer. Lung cancer looks different from liver cancer. Each cancer type leaves a specific identifiable molecular signature in your blood. Not just cancer is here, but this specific type of cancer is here, and it's probably coming from this organ. This is the biological foundation for something that until very recently would have sounded like science fiction. A single blood test, one draw, that can detect over 50 cancer types simultaneously and tell you where in the body to look. It's called the Galleri test. The machine that reads the signal is remarkable, but detecting it is a different kind of challenge entirely. The circulating tumor DNA in a blood sample is fragmented, short, degraded pieces of genetic material. The cancer signal in early-stage disease is faint, and the methylation differences that distinguish pancreatic cancer from ovarian cancer from lung cancer aren't simple on-off switches. They're patterns spread across thousands of positions simultaneously. This isn't something a human can read. It requires machine learning. A company called Grail spent years building AI models trained on thousands of samples, cancer patients across 50-plus cancer types, healthy controls, early stages, late stages. The models learn to recognize the methylation signature of each cancer type from the fragmented, noisy signal in a blood draw. The result? A test that outputs either a clean negative, no cancer signal detected, or a positive with a predicted tissue of origin. Not just something's wrong, but something's wrong and it's probably coming from here. That second part, the tissue of origin prediction, is what makes this clinically usable. Without it, a positive result means scanning the entire body. With it, doctors know exactly where to look. Targeted follow-up, efficient investigation, real-world medicine. In a study of patients presenting to doctors with possible cancer symptoms, the Galleri test correctly detected a cancer signal in approximately 2 out of every 3 people later confirmed to have had cancer, and in 85% of positive cases, it correctly identified the tissue of origin. For 47 of those detectable cancer types, the ones with no routine screening program anywhere, this was the first systematic early detection approach ever evaluated in clinical practice. But that was a symptomatic population, people who were already worried, already showing signs. The real test, the one that matters most, was whether this could work in people who felt completely fine. So Grail and the NHS did something extraordinary. They enrolled 142,000 healthy volunteers, gave them annual blood draws, waited, and on May 30th, 2026, at the world's largest oncology conference, they revealed what they found. In Part 2, the trial results, what the blood knew before the body did, and what this means for the future of cancer medicine.